Anjali Venkateshwaran, Senior Director of Marketing at Biomere, and it is my pleasure to introduce Dr. Hannah Sheehan, who will be joining me today. Hannah is the CEO and CSO of SauveBio, that is a startup focused on flow cytometry and cell sorting. Biomere has recently partnered with SauveBio to offer cell sorting on samples collected from in vivo studies. Prior to founding SauveBio, Hannah developed complex flow cytometry assays at AbbVie. She received her PhD in biology from Northeastern University and her bachelor's degree from the University of Rhode Island. Joine Biomere is a preclinical CRO with locations in Worcester, Mass., Richmond, California, and multiple sites in China. The company's portfolio includes comprehensive PK/PD and toxicology services, and you can learn more about Biomere's service offering at www.biomere.com. So let's get into today's topic on complex flow cytometry and cell sorting. Hannah, I'm so glad you could join me today. I'm so excited. So let's start with an overview of your journey. How did you think of founding SauveBio? What were the driving forces to move from pharma to your own company? So that seems like an amazing journey you're on.
Thank you, Anjali. And first off, thank you for having me. I'm really appreciative of being here. I look forward to speaking with you in detail today. So the mission to start SauveBio was really founded on 10 plus years experience in high parameter and advanced flow cytometry, as well as cell sorting. And we had noticed a critical threshold in the field where the science was advancing at a rapid pace, but to develop these methods in-house at a lot of different companies just requires a massive amount of capital, as well as very specialized staff with a really specific skill set of expertise. And this isn't always feasible for smaller research companies. So we sought out to form a company that was focused on being an expert service provider that could serve as an extension of people's research team to develop these services in a way that was more effective and got to the point a lot quicker so that science could advance at the pace that it was capable of advancing.
Yeah, and that's really interesting, right? Because if you think about it, flow is a very important readout or cell sorting. It's one of the key steps to downstream analysis. For example, you sort out different populations and then you get these pure populations and analyze them. But I think that would be a tricky thing to have full time, to have it in-house, because much rather you go to the experts. They provide you basically with the data in the case of flow or actual sorted cell populations, and then they take it and go on their merry way to do whatever analysis. So I think, and so I'm curious, in your opinion, have there been other companies who've done this kind of platform, or is this kind of something a little bit like more novel where you're like, hey, there's nobody out there doing this, we should be doing this?
Yeah, so when we thought about forming the company, we started to look at the market and see where other CROs offering this service. And the answer is that they were
Hmm.
mostly for analysis, not really the cell sorting component.
Right.
But what was happening was that this work was being done at the technician level, reasonably so, where it didn't really make sense for CROs to be hiring PhD level scientists to perform
Yeah.
this work. With 10 plus years experience or even more, so
Right.
it was not feasible cost wise for CROs to offer it in the way that we felt it needed to be offered. So given that it's such an advanced skill set and that the capital to get the facilities up
Mhm.
and running is so high, it just made sense to be a dedicated service provider. And as I wanted to form a CRO, I knew that this was my main area of expertise. So it
Yeah.
doesn't make sense necessarily for me to be offering a wide range of skills here when flow cytometry is the one that I can deliver at the highest level. So this
Makes
was a
sense.
need in the market for sure.
I think that makes a ton of sense, right? And I just want to pause here because when people say flow cytometry, right? I think sometimes there's a little bit of a misconception, like, oh yeah, you have good antibodies, boom, you can get gorgeous flow data, and you see all these, beautiful scatter plots, and you see all these data, and it's like, but flow is tricky. It is
Absolutely.
tricky. I think it's a tricky, assay, because there's so many things, and that you've parameters you've got to account for, right? And especially not just the antibody part of it, your input samples, like you and I have talked about in the past about sticky cells, like
Yes.
skin cells. We've had conversations where it's like, oh gosh, that's just clogville, happening. And I think that's something to be said. So, maybe we'll just that's a segue into talking about maybe a little bit about the complexity of flow and because I know you've done a lot of really difficult, tricky assays. So your viewpoint would be super refreshing on educating our listeners on, the complexities of flow.
There's so much to it. So any one experiment is going to have a number of parameters that you need to be focused on. So not only is it identifying the markers that you're interested in looking at, you also need to assign fluorophores or fluorescent particles to these molecules that you're interested in looking at in such a way that they match the expression level. So you need a brighter fluorophore with your least abundant protein. So it's really important that you set up the panel in the right way in that way. But then, as you mentioned, there's other considerations like sample type. So if you're working with a tissue, or skin in that example, there are a lot of different complexities for how you should prepare those samples ahead of time before even staining them, just for the dissociation of the tissue, so that you can end up with the highest viability cells down the line, and that the marker expression you detect is accurate and that there's not a lot of autofluorescence or other spectral issues that can come up. And
Right.
then it's designing these panels with a high number of fluorophores and markers. So there's a lot of considerations there where not only does it have to match the actual expression in the tissue itself, but it also really needs to match the goals of the experiment. And the
Yeah.
reagents available for each of these things are not always perfect. So there is a little bit of trial and error there. So by
Yeah.
working with somebody that has a ton of experience in optimizing these panels and in assay development in particular, you end up skipping a lot of that troubleshooting
Yeah.
phase where if this work is done by somebody that just doesn't, have any experience with that one tissue type or with those markers, you might not get the results that you're looking for in the
Yeah.
end. And it can be a huge cost, and it can also be a big waste of time. And then there's all other issues that come into play there where you might not necessarily, as the flow cytometry operator, be able to tell that anything went wrong, except for if you have
Oh.
expertise. So it's, you get out of an experiment what you put in. And
Hmm.
sometimes you might think that you've isolated the cell population of interest for your RNA-seq analysis or something similar to that down the line. And then you get back the analysis and all these expression markers are, found that shouldn't be there in that sample type. And you realize that maybe what you isolated isn't what you thought at all. So that can be a huge issue for
Right.
researchers. So knowing
Yeah.
what you're working with and knowing how best to approach what your goal is a really fine-tuned skill set that I think takes years and years to develop.
And I think you raise a really good point, right? Because when people, when CROs, when people when people outsource flow to CROs, right? CROs, I've been in the CRO business for a long, long time. It's like you get it, you've got an SOP, you've got, you just there's a deliverable at the end of it with a certain time frame. And I think what you're referencing is method development, right? And that's where I think Sauve has this edge because you can do that method development. If it's like a very unique sample type, as we talked about, or a very tricky cell surface target that's very, fragile, labile, degrades, whatever it is. I think that method development concept is so critical because once you've got it and you've locked in your protocol and you've moved it to an SOP, then you're in business. But that method
Right.
development can get incredibly tricky. No, I absolutely, I see your point, and it makes so much sense that this is what you're focusing on. But let's jump to cell sorting because that's such an interesting space for me, and I know there's just tons of applications. I mean, the first thing that
Yeah.
pops into my head when I think cell sorting is immune cell profiling, right? Or in immune oncology, you treat, with some kind of a checkpoint inhibitor or some kind of an oncology, whatever, an immune oncology therapy, and you're like, hey, what happened to my immune cell population mix? Did this go up? Did that go down? But I'm sure there's other, there's a gazillion probably applications for cell sorting. So, yeah, what's your opinion? I mean, from sitting from where you are as an expert in cell sorting, where do you see the fastest growing areas of cell sorting, and any cool new emerging areas?
Definitely. So one that I would say that you kind of are mentioning here is cell and gene therapy. And this is a massive area that we're working in where we can really help to, develop the understanding of what you're seeing. So what immune cells were activated by a treatment condition, for example.
Mhm. Mhm.
Was the tumor actually depleted? The tumor cells, were they
Right.
depleted from the host? Other things, such as were there off-target effects? This can all be
Right.
analyzed by flow cytometry. So within one experiment, you can get a much more complex image of what has happened, based off of that treatment. Another area that I find really interesting is single cell omics work. And this is
Okay.
definitely a growing area in the field. So we are isolating pure populations for single cell RNA-seq analysis and proteomics and attack-seq. And these things, you can do all of them without sorting cells, of course, but sorting cells gives you a much more pure population to look at. So rather than having a tissue that has a ton of different cell types and then, looking just at the one that you care about, we now can make sure that the analysis is only based on that one cell type that you care about. And then you get a much more complex depth of coverage on the analysis there. A third one that we actually are not working in right now, but that I wanted to mention as an emerging area is complex microbiology applications.
Oh, okay.
So we aren't doing bacterial isolation by flow cytometry right now, but we do plan to eventually get a dedicated instrument to do this work. And when I say dedicated instrument, that's the hint as to why we aren't doing it currently.
Right.
If I'm going to give my customers the best cell populations, I cannot ever run bacteria through that same instrument and make sure
Mm.
that the
Yep.
mammalian cells never get contaminated. So we're going to get a dedicated instrument for that down the line. But it is really interesting to think of not only analyzing mammalian cells and isolating them by flow cytometry, but also developing our collective understanding of the microbiome with
Interesting.
smaller, bacterial subsets. So that's something that I think is really interesting.
That's fascinating. I did not know that because when you think microbiome, you immediately jump to 16S metagenomic sequencing, where you basically then build out the very complicated tree of all the different genus and species of bacteria. But that's fascinating. That you'd be that's definitely a very, very interesting area, and with the increase in interest with the increasing role of the gut microbiome in this whole, I mean, you think of the gut brain axis, that's like one of the big ones, but I'm sure there's so many others where more and more people are finding out how important how informative the biomicrobiome is as a biomarker is for disease state, for therapeutic response, etc. That could be a really, I see that as a very cool emerging area.
I think so
Interesting.
too. I think it's something that's really needed in the field where maybe for
Yeah.
for new compounds that are developed, we should always be looking at the effect that they have on the microbiome within the body.
Yes.
Or at least most of the time it might make sense where it's something that we're missing in the analysis right now, particularly because a lot of microbiologists don't have a partner to work with for this work.
Yes.
CROs, just like me right now, don't want to put the cells through the machine. So
Right.
in that way, we are limited. But given that SauveBio plans to add the capabilities to do this, we will be one of the partners within the Massachusetts area that's capable of helping them with this
Yeah.
work.
And that's interesting, right? Because jumping kind of, I was just kind of, as you were talking about the microbiome, I was thinking about, Biomere, because we do in vivo work. So, rodent, large animal models. And, you know, th again, that would be really interesting because right now we don't even look at the microbiome in any
Right.
way, shape, or form. But that would be really interesting. So if again you mentioned, even if it was a simple oral small molecule, right? Is because we all know what antibiotics can have a very detrimental effect on the gut microbiome over time, and there's a lot of things associated with issues associated with that. But that could be something really cool, and I think it just the other thing it also does in a interesting way. And I just want to bring this up, not go too much down that tangent, but it has a huge impact on the 3 Rs because one of the things is when people still use animal models, right? And they, I think they'll continue to use it in some way, shape, or form for the foreseeable future. But think about it, if from one animal you can get so much data, you can get the microbiome response, you can get, of course, your standard biodistribution, look at PK/PD, you can look at toxicology. It's actually so aligned with the 3 Rs where you're getting so much data from a single animal and that you're not you're basically getting almost close to complete in systemic story which I think has a ton of value. And you're right. I think people should continue looking at the gut microbiome because of it seems to have its fingers in so many different, physiological pies as it were.
Yeah, a
But
mouse
one,
can't tell you that the
yeah,
stomach hurts, So, given that we don't necessarily know symptoms,
right,
it's
right.
nice to actually get some sort of a complex view, by measuring distinct parameters with the bacteria in the microbiome, in the gut
Yes.
microbiome, to really understand that maybe when we do put this compound into humans, that there would be something that we need to consider.
Absolutely, absolutely. And that also may help with patient segmentation. So certain patient populations may have to be excluded, what have you. I think fundamentally, though, I think we get all in agreement that the more data we can get preclinically to inform before you go first in human, the better. There's no such thing as too much data in my
Yeah,
mind when you think of preclinic, right? Go into clinical trials as informed as possible. It's you're increasing your probability for success.
definitely. I
So
agree.
I'm gonna segue, Hannah, because my this when we first met and we were talking, you always think sorting equals cells. And that's kind of
Mhm.
like almost such a simple way of looking at it. But then, after doing some more research and talking to you and whatnot, it's not just confined to cells. We're looking at, you mentioned, I knew of exosomes and vesicles, and then you mentioned nuclei. And I was like, oh gosh, we're talking subcellular organelles, we're talking like vesicles of different sizes. So tell me more about how, non-cell sorting rather, or sorting of non-cells is accomplished, and what are the kind of the technological advancements, or how is it done, and what are the requirements for that?
Yeah, so nanoscale sorting is a really niche area, and this is something I'm super excited to talk to you about. So, exosomes, for example, were within the noise region. So at the lower limit of detection on a flow cytometer, you just have too much noise where you can't decipher small particles from that noise. So just, for decades, that was the limitation essentially. And then over time, what we've done is developed better lasers, so higher power lasers, particularly in the 405 nanometer laser range, which we call our violet laser, would be one. We've also developed better detectors for the signal from those lasers, and that's been really important, including avalanche photodiode detectors, as well as advanced PMTs, which is what my
Hmm.
instrument has, my BD Symphony S6 primarily.
Hmm.
And last but not least, thresholding and triggering within the system. So we used to use the size of the particle as the only way that we were deciding whether it would be considered an
Right,
event or not. But now
right.
we are able to use a fluorescent signal for that. So given that exosomes are extremely small, size is not something that we really can use as the trigger to decide if an event is worth pursuing. But now that we can use a fluorescent signal, we can use something like a lipid dye to be able to, get a very small signal from these exosomes, but to then amplify that signal and be able to actually use it. So this is a
Yep.
development that has allowed a whole new avenue of research, really, where we can isolate exosomes and, even just analyze them by flow cytometry. And I've actually been really involved in methods development for nanoscale applications over the course of my career. So I wanted to mention one that not a lot of people talk about, which I was involved in the development of mitochondrial isolation by flow
Right,
cytometry.
right,
Call this
right.
fluorescence activated mitochondria sorting, and it was developed in Dory Woods and John Tilley's lab at Northeastern, and I was on that project. So I ended up getting to write a methods textbook chapter and a few papers about
Nice.
the method, which was really exciting during my doctoral studies. And this is a really interesting area of research as well, where people now can kind of get a better image of submicron populations of mitochondria. So within the cell, mitochondria are not all the same. So they
Right.
have distinct functions, and some are respiring more or less, and some are driving different cellular phenotypes than others. So it's really important to get more of an image of, I guess, mitochondrial subpopulations within the cell. And now we have a method to be able to do that using flow cytometry. We also can isolate mitochondria and then be able to use them in therapeutics. So there are some labs that are actually using mitochondrial injection back into a host to
Right.
drive, certain phenotypes. So people
And
use it
is
for
that when,
oh,
sorry, so let me ask you a couple of questions here. So is that when it's like mitochondria exhausted and there's like metabolic exhaustion, so then you can kind of like uh, you know introduce more invigorated, probably mitochondria.
yeah.
But one question, and this is a technical question, because like when you think of cell sorting, right? That you're basically looking at you're sorting based on cell surface markers. That okay, if you express, receptor A, you're this population. I'm simply oversimplifying, but just for the purpose of illustration. How do you do that for mitochondria? Are there specific markers on the
There are.
mitochondrial membrane that allow sorting, or and do these change with different physiological states?
They do, yeah. So one example would be mitophagy. So this is the cell's
Okay.
process to get rid of defective mitochondria that are typically no longer respiring, and that can build up within cells and cause damage. So really you do want to get rid of them. So there are proteins called PINK1 and parkin that end up
Yeah.
externalized on the outside of mitochondria, and parkin being one that drives Parkinson's disease,
Right.
actually.
Yes.
So yeah, so this is something really interesting where mitophagy, happens within the cell, but then we can use this to isolate populations of mitochondria since it's on the outside of the organelle. That
Right.
means without permeabilizing the mitochondria, we are able to use use antibodies that are against those proteins to be able to then isolate the subpopulations of interest. So if I want to say, what is the difference, during proteomic analysis of
Right.
mitochondria that either are in mitophagy or aren't, I can now use antibodies to
Right.
isolate populations of mitochondria and try to understand what's happening there at a deeper level by running proteomic analysis downstream.
And even potentially rescue, right? Because I think even pink one, along with Parkin, is part of the Parkinson's disease targets. And I mean, just maybe thinking a little too crazy out there, but that would be so interesting if you could find a way to, dry to clean up, vacuum up the defective mitochondria, and maybe that has a better impact on, the viability and the health of the neurons that are impacted in Parkinson's disease. And that's really going into the root cause, right? And not just because right now, right, people take L-DOPA, levodopa, whatever, and over time they get, it really kind of you lose sensitivity to that. But this is really going at it at a very, very deep level and fascinating stuff. No, yeah, might have had. I didn't realize that. The mitochondria had such a nice mitochondrial membrane cell expression. Fascinating
So
stuff. My
certain proteins
goodness.
they do and certain ones they don't, but we
Okay.
actually had figured out how to permeabilize the mitochondria as well. So
Okay.
markers that were within the inner membrane, even we were able to, and matrix
Wow.
actually, we were able to use for isolation.
But without kind of lysing the mitochondria and releasing all the contents, you were able to get to the inner membrane.
Mhm.
That's
Yeah,
pretty impressive. That's
yeah,
a technical skill. Because you
yeah,
basically you
and
don't I visualize the mitochondria exploding, going kaboom, and all the contents spilling out, and then you're kind of done.
yeah, I mean, we definitely had some trial experiments that went that way, but
I bet.
once the protocol was optimized, we were able
Yeah,
to detect expression, sufficiently, really. So it was very
right.
exciting. And in that case, mitophagy was involved in age-related infertility in women, which was what we were trying to understand during those experiments.
Interesting.
So you see mitophagy defects in older women who are now, not able to get rid of mitochondria that are causing problems within their body. So there
Interesting.
are a lot of ongoing studies for that, but basically we shouldn't even think of mitochondria as only being important for Parkinson's because, cancer
Right.
is by mitochondria,
Yes.
fertility problems,
Yes.
and many other things. So, that's the mitochondrial example. Exosomes is a whole nother rabbit hole that we could
Yes.
go down. But really,
Right.
organelles are driving so many important cellular phenotypes. And this is why it's so important that we have methods to be able to study these things. And these
Yes.
are methods that other CROs, to my knowledge, very, very many of them are not offering, just because this is a really, really, niche skill set. So the
And
fact that
it's
SauveBio is offering
tricky.
that,
It sounds really
definitely.
tricky, and that's coming back to what we started with, that on it makes sense. Like think about it, you're like, yep, this makes sense. But executing it, I think, is a whole different other level of trickiness, and you've got to have some really specialized skills. And as you said, trial and error, figuring out, okay, and locking in those protocols, locking in those little, the secret sauce tips and tricks. No, and
There were
you're so right,
yeah, that had
like.
worked on it. Yeah,
Right.
so
And,
for people were working on that before I was even there. So,
right.
the fact that I was on, quote unquote, the cutting edge of developing that method, I still was coming in after years of troubleshooting had
Right.
already happened. So I
Right.
think it's really important to note, because this expertise does not just come from me. I have this network of people that have taught me over the course of my career, and I'm using that information to be able to better serve
Right.
my clients now. So I think if you're new at flow cytometry, you can do basic analysis just fine. It really doesn't take extremely long to get that up and running, but then to do these fine-tuned applications and isolation of rare stem cell populations and things like
Right.
that, you just
Right.
really do need to have somebody who has that skill set.
I agree with you. And I'll also say, based on what we're seeing, the low-hanging fruit of the easy, straightforward assays, that's going into the minority. People still do a little bit of, some kind of some simpler assays, but I think more and more and more there is this trend towards more complex assays, more complex study designs, looking at, as you said, rare populations. For a while, I think there was this huge buzz for gamma delta T cells, which are like what, less than 1% of total T cells. Like I was trying to visualize, oh my God, the cell sorting of that must be one of the most incredibly tricky and complex assays. No, you're absolutely right. I do believe though, like, one thing I want to think about is, we all studied like cell biology in school and college, right? We all have the structure of the cell in our head and we know, the ER, the Golgi, the mitochondria, and all the different and, cytoplasm, all the different organelles. But I think this kind of cutting edge technology and technology allows for us to almost revise, like update, change our perception of what these organelles do, right? A lot of people say,
Absolutely.
"Oh yeah, cell surface. Oh yeah, if I can just basically like go to the cell, attach to the cell of interest, I'm good." But actually, it's what happens inside the cell, the very complex cellular machinery, I think. And it's really interesting as you giving, the example that you gave of mitochondria and mitophagy. I mean, I can just see that happening with other organelles. Who knows? Maybe people figure out how to do, sorting on Golgi. Who knows? It could
Right.
be
And also
completely.
there's this whole field developing of, and I actually had a recent paper about this, but so the way that different organelles interact with each other. So we used the
Yes.
mitochondrial isolation method along with exosome staining dyes to then
Okay.
see that there were mitochondria contained within exosomes in these samples. So then
Right. Oh, yeah.
well, maybe it's not that there's anything wrong with either of the two organelles separately, but their interaction over time might be altered. And
Yes.
now we need to understand
Yes.
that. So I just think that if the basic analysis methods for the cell surface were sufficient, we would have cured more things by now. So,
Yeah,
to develop these methods
I
in such a
agree.
way that it serves the science is so important. And that's why SauveBio wants to be on the cutting edge of this work. And we
Right.
really hope to be an excellent partner for our research customers doing this work.
That completely makes sense, and one of the themes here that's emerged is the complexity. So I'm gonna ping you with two questions. We can start. The first question is instrumentation. So there's a lot of cell sorters available commercially, and seems like that whole instrumentation development platform, they're just developing, and more bells and whistles, incredible sensitivity, like the machines are just getting better and better and better. So in your opinion, you know my first question is around instrumentation. So basically, in your opinion, what are the cell sorters in the market, and they all have different pros and cons, I'm sure. And I want to ask you why you picked on the Fax Symphony and what you think, how that can be used for the work that you're doing. Second one, which we'll talk about a little bit later, is the big, big, big question of data analysis. So those are the
Right.
two questions, but let's start first with the instrumentation question.
Sure. Sure. Instrumentation is a really important part of beginning your research journey for these questions. So I think it's really important to talk about. And in general, the sorters that are used today are very similar in a lot of ways, but have kind of minute differences that are really important to understand. So in general, FACS is the concept that they're centered around, where you have a stream that you're doing hydrodynamic focusing, where the cells are then put in a single file line, essentially. And then what happens is that they are interrogated by a laser, and then signal is collected by detectors. Then if it is a cell that you would like to isolate, you are going to apply a charge essentially to that stream
Okay.
at an exact point so that that cell can be pulled, into a collection tube, and then everything else will go into the waste. So that's just the
Yeah.
general concept. But the instruments, they range in complexity. So as far as parameters that can be analyzed at once, most sorters can do 12 to 14 colors at a time. So 12 to 14 markers. But that's kind of at the high level of their capabilities versus the Fact Symphony S6, which we decided to go with, can do 30 plus parameters at a time. So, I might have to, if I had a different machine, I might need to do a larger panel to identify a small subset of cells that I'm interested in and do that just by analysis on a different machine, and then simplify that panel for use on a sorter. But now I'm able to do really complex analysis with high numbers of parameters, and then use the sorting capabilities to isolate my subset of cells all in one experiment. So I can
Right.
generate really complex analysis data at the same time as sorting populations of interest, rather than needing it to be a two-step process. There are also other considerations like how gentle is the machine on the cells,
Mm.
and the Fact Symphony S6 is really great for this, but there are some machines that are microfluidic sorters that might be even more gentle. So for very specific applications, that could be necessary as well. But that's the main gist of it. The machines really just range in how fast they can sort, how complex the samples that you can sort, or really like what the labels are on the cells and how complex the panel can be. And yeah, so how gentle, the speed, and complexity, I would say, are the three there.
And you mentioned microfluidics and things like that. So, what do you think? I mean, you've got to strike that balance, right? Because you don't want a machine that's very, very sensitive, but runs into clogging issues or has some maintenance issues. So, in my mind, I think that there's a balance there, right? As there is with almost any like omics or any kind of a analysis platform. I'd love to get really, really specific and really, really precise, but am I introducing, reproducibility issues? Am I introducing technical clogs, jams,
Yeah.
fixes, machine, the
And
the
the machine's
calibration
only
getting
part of
thrown
that.
off, stuff like that.
Right. Yeah, so the machine is only a part of that dynamic, really. So, I can have a great machine that is known for not having a lot of clogs, but if I still don't have the expertise to set up my experiment in such
Right.
a way that I'm preventing those clogs, I might still run into that problem.
Yeah.
So
Yeah.
it's identifying an instrument that's effective for what you're trying to do, actually using that instrument if it's available to you, and then also having the expertise to prepare the samples in such a way that you reduce issues. You're keeping thresholds or event rate low, rather. And that's really important. So you want to be sorting as fast as you can to keep the cells viable without going
Right.
so fast that you're causing issues in the actual isolation of the
Yeah.
cells that you're interested in,
Yeah.
or at such a high pressure that you're killing the cells upon isolation. So there's just this really intricate balance, I would say.
Yes. And and that's an interesting concept, right? Because when labs want to purchase a fax machine, it's so easy to get, for want of a better word, seduced by like, oh, you could do this, oh, you could do that. You can, the features are amazing, right? And then it's a good, I think it's a good practice to think and pause saying, what am I actually going to use this machine for? What am I trying to get out of it? Because I actually know of a couple of instances where, a very, very high-end machine was purchased, but it was overkill. It was kind of like you bought a Canon when a maybe like something much smaller would have been much easier. Like a pistol was a
Exactly.
cannon. I mean, it was something that pops into my mind. But and I think that's where, expertise like yours comes in, which is, okay, what do I want to do? Is this going to help me get there? Answer is yes, done. Whereas opposed to, you spend a ton of money because these machines aren't cheap, but not by a long
Right.
shot,
Oh, yeah.
right?
They can go upwards of $1 million. So I think
Right.
it's really important to, as you're saying, identify the machine that isn't overkill but meets your needs. And this is where things can get tricky. And this is why I think outsourcing can be a great option,
Yes.
particularly for smaller biotechs, where it doesn't make sense to buy a Fact Symphony
No,
S6.
no.
Might need it compared to your other instrument for one experiment, but you might never need it again. So to outsource that work to somebody
Yeah.
like me, that I'm building out this lab that I can now meet, a wide range of capabilities. So right now
Yes.
it's the Symphony S6, but then I'll add on other instruments that have other strength points so
Yes.
that I can meet any customer needs. And in that way, you just reduce the amount of downtime on the instrument, I would say, and
Yes.
really within labs and how much labs cost to keep up and running now, it just makes a lot more sense to, I guess, outsource the work and make it so that there's no reason to bring that
Yeah.
in-house and just have it be wasted.
This is actually in complete parallel to why, people outsource in vivo studies, right? Because you don't want to build a vivarium, hire the staff, maintain all the standards, make sure that everything is all the paperwork or regulatory requirements, all that is met when somebody else already has this set up and, has basically the infrastructure and the personnel in
Right.
place. And I think there's a case to be made for that because, as you said, the only reason I would see somebody doing this in-house is if they're doing it literally every day. But if you've got like a few programs, drug development programs, and you need like sporadic, like enough data, I need to do this set of studies to get to my next milestone, whatever that, drug development milestone is, it totally makes sense to just pair up and collaborate and outsource with the experts, get the data and keep on going because there's such a need for speed to be first to market.
Definitely. This is
It
all
makes sense.
race, you know.
Yeah.
Yeah, people
Yeah.
are racing to, meeting those milestones against their
Yes.
competitors. And it takes you 6 months to a year to set up a flow cytometry facility
Exactly.
for one experiment that you need replicated 3 times. I could have
Doesn't
that
make
done
sense.
within a few weeks versus, take
Exactly.
that time to get it set up. And the capital
Exactly.
is extremely different
I
as well.
make Makes sense. And now, then, okay, so then we're going to go to my second question, which is, I think, my favorite question, data analysis. Because, with any platform, right? And as, whether it's omics, whether it's facts, whatever it may be, data analysis is critical because at the end of the day, you've got to be able to analyze your data, come up with actionable insights, and take that information and move it forward, right? And a lot of, as you know, a lot of the, clients that Biomere works with and Solve works with are biologists, right? These are folks who may be chemists. Their focus is on developing their therapeutic, right? And
Yeah,
so for them, um, it's hard it's hard to have the skills to analyze the data, right? Like, okay, what does
right.
this mean? I got all tons and tons of raw data. As you mentioned, you can get a lot of data from one experiment. Okay, how does it make
Right.
sense? And that's where I think a lot of people get tripped up because it's like, okay,
Yeah,
this is really cool, but what does this mean? So,
definitely.
I think your expertise there can add a lot of value.
Yeah, so a lot of times people are getting raw files back from research partners, including CROs. And this is essentially useless if you don't know how to use the information and analyze it
Right.
effectively. So, for example, with a 30 plus parameter experiment, you can't just do manual gating and have this be
Mm,
logical because the human eye just can't, understand the complexity of 30 dimensional data. So we use algorithms like tsne and umap to do dimensionality reduction and be able to then group the cells in the analysis within, I guess, clusters is how you would refer to it. And within those clusters, there are expression markers that are associated, and that's why it was clustered in that way. You can also assign statistical power to that. And then we use things like flowsum algorithms to just further expand our understanding of how many exact distinct populations are within that data set. And it also removes human bias in that algorithm. So that's really
yeah.
useful in a lot of these. We also then are generating data analysis reports. So we're not just giving you back raw files. We're doing all of the FlowJo analysis, which is the typical platform that's used for flow cytometry data. There are others as well, but that's really the workhorse, I would say. So we're generating that data. We're then putting it into a PowerPoint format and presenting it to our customers so that they can not only see the conclusions all in the FlowJo analysis, but they then can understand how we got to those conclusions,
Right.
what we think
Right.
that they mean, and how we believe that you should move forward from here. So it just
Right.
takes the guesswork out for customers that don't have
Yeah.
familiarity with cytometry. Because if I were to receive a 30 parameter analysis report and nobody walked me through it, I don't know
Right.
that I would necessarily understand
No,
it if it's not my
absolutely.
either. So we really serve as an extension of the research team by, talking in depth with our customers and helping to suggest next steps and to troubleshoot things and move forward in an effective way. And it's, I think, unbeatable compared to some of our competitors in that space.
Completely. And the other thing I was just thinking is also this is so useful to get like you get the summary. You have all the raw data if if you're inclined to review it, so knock yourself out. But and
Yeah.
then that combined with other approaches, right? Because a lot of times, people will do flow, they'll do ELISA, they'll do whatever it is, different endpoints, and then you put it all together, and that's when you have a much more complete story. So, and then it's like, oh, okay, and it's really, I think the perfect study is almost when one method confirms findings from another method, and there's this whole orthogonal confirmation thing going on, which is wonderful because then there's a confidence that, okay, my drug, in the best case scenario, my drug, I put it in there, it did what it was supposed to do. It disrupted this. This is the data. This is one thing I need to be watching out for in terms of, off-target effects. And then you can actually go downstream and start moving towards, say maybe even IND enabling studies, what have you. I think that's a key bit, right? Is making it easy so that folks don't have to start becoming experts in spaces that, you know, are new for them. And I think that's the challenging part because if you talk to drug developers these days, right? In the old days, when people did small molecule analysis, it was very kind of structured and you just went down the path and, it was, and then you got to IND. But now I think with all these complex assays, with the data analysis has become insanely complex. The methods are
Mhm.
insanely complex, very, very informative. I do feel sometimes that a lot of drug developers are drowning in the data, or they just, or, even in a quagmire trying to figure out, okay, where do I go? What do I do? Which direction do I to move forward in? So I think, this concept of partnering with people who do specific aspects of drug development very, very well, whether, you're looking at in vivo, you're looking at biodistribution, you're looking at, you know, um, changes in cell populations, you're looking for, we talked about mitochondrial, changes in physiology and function. I think that's such a smart way to go about doing drug development because this way you're tapping into all these resources. And in my mind,
Mhm.
drug development should not be a linear process. It should be an
Right.
ecosystem, right? Where
It can't
you,
be linear or it would be easy. And it's not easy. So
right?
that doesn't make sense. When we talk about people becoming experts in one method, so in this case, flow cytometry data analysis,
Yeah,
but that's really just not what's even possible. So say you get
no,
a data set back and you need to make actions based on that data set.
right.
Even if you spend a few weeks or a few months on Google to try to become an expert in this,
Yes,
how do I now know I'm an expert enough to make the conclusions I'm making when it's something completely new to me? So it's
right.
just you're expecting things of people that are risky, I would say, and difficult and challenging, but also really risky. And I think that that's a huge part of it, where I
Yes,
might think that I fully understand the data set that I'm trying to make interpretations on right now. But maybe I still don't because somebody
right.
else out there with 10, 20 years experience in the field is
Yes,
able to understand it at a deeper level and
absolutely.
is saying something different than I think. And that's a real risk in the field where you might then have your
Yes,
program go in a totally different direction than it should.
yeah,
So it's really important to partner with people like Biomere and SauveBio that have, fine areas of expertise that are,
absolutely.
really, really well tuned, I would say.
And, the last thing I'll just say to conclude that thought is, and also people who've done the work, who've got the real world experience, right? Because, yes, there's a lot of like information out there. You can get a ton of theoretical information, but I know it's a case with you, and I know it's a case with the biomedical scientists, where on paper it makes sense, but you're like, wait a minute, no, 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 no. I've done this before, and you better watch out for X because this is going to happen. And that is that priceless real world experience, right? Where you've done it, you're like, okay, no, 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 this looks good on paper. Reality may be a little bit different. And that's that kind of, it's priceless. I think that experience, that expertise, is truly what, is the value proposition,
Absolutely.
for people to outsource, especially for you, people being able to access your knowledge, your expertise. I mean, that's a very compelling value proposition. Same with, at Biomere, people are able to access our scientists' expertise, the history, the track record. Like we've done this before, it didn't work, and this is why. And as you said, stops people from going down the wrong path or going around in circles or what have you. I think that's absolutely priceless. So wonderful. Well, on that note, Hannah, Thank you so much for sharing your insights. I mean, I've enjoyed it. I think the time's just flown by, but and I know we could talk for hours on this. And for our listeners, I'll say the Biomere team is so excited to partner with Hannah and the SauveBio team because I think we can bring, together we can bring so much value to maximize the actionable data, the concept of actionable data from these studies, so you can, as a drug developer, can move forward and you're not kind of, staying where you are. So if you're interested in learning more, you can check out SauveBio's website. It's www.sauvebio.com. And if you want to check out Biomere's services online, you can go to www.biomere.com or email us at bd@biomere.com. So with that, I will say thank you, Hannah. This was a wonderful talking to you. And on behalf of Biomere, I say we really look forward to this collaboration.
Thank you. We're looking forward to it as well. And we really, really look forward to driving discovery with you.
Wonderful. Thank you so much.